Hello and welcome to my bioinformatics tool presentation. I will be talking about the S-plot2 tool, how to install and how to use this tool. Now firstly I will explain to you what the different uses are for the S-plot tool, then I will go over how to install this tool on your desktop, mostly with the Linux OS system. Then I will go over the different features that are in the program, and lastly I will pose a hypothetical scientific question and answer this using the S-plot2 tool. Now firstly, the S-plot2 tool is a program that allows for rapid visual and statistic analysis of genomic sequences. The tool is used mostly to compare two different bacterial genomes by generating a two-dimensional heat map where we can find the similarities and dissimilarities for two given genomes. The dissimilarities can be interesting because we can investigate if they harbor genes that are required by lateral gene transfer, or if there were any insertions or deletions of specific genes. Now, over to the installation. First things first, you have to download Java and install Java on your system, because otherwise the S-plot2 program will not work. Once that is done, you can download the S-plot 2.1 jar that can be found in the link of this YouTube video or in the leho link that is also in the description. When you have downloaded this file, you can go to the directory where you have downloaded it and then execute the jar file by typing in Java --jar S-plot 2.1. Now over to the different information of the program. First things first, we have the insertion of information. This can be whole genomes or just genes. You have the ability in the program to customly label your axis and also insert the files in your X and Y axis. Note that every file that is inserted must have the suffix extension FNA. This is just a different extension that also refers to a FASTA file. Now over to different parameters. For the parameters we have the k-mer size, the genome window size and offset size. Now first, the k-mer size is typically referred to as the possibilities that a substring can have with a length of with a supposed length. The k-mer size mustn't be small, too small, because then a lot of information gets lost, because the bigger your k-mer size is, the more information it will contain, but it mustn't also be too big, because if you have two sequences that are widely different from each other, then you will have a low correlation value to get any info out of the heat map. Now for the genome window size, I will elaborate a bit more. Now suppose you have this sequence and you choose to have a window size of 10, then it will calculate each k-mer frequency for this blue block. Now with the offset, if you put an offset of 5, it will take the beginning of the blue block, go 5 nucleotides, and then calculate the k-mer size also for this green block. So with a window size of 10 and an offset that's 5, the k-mers will be calculated for both the blue and the green blocks. Now over to the analysis or result tab. This is an example of how a result will be shown. The different parameters that you have inserted will be shown in the top. The correlation scale will be shown on your right side, with 1 being black and 50% correlation being green. Then you also have your labels, where you have your customly named axis, and also how much base pairs one pixel is, or one window is. Then I don't think you can see it that much, but here there is a black line, and this black line represents alignment. So regions with high correlation, that's why they are black. Then you also have regions with low correlation, so these green regions over here, it's very clear. These can be high conserved genes or genomic islands. Then we also have an example of an rRNA. This is quite rare because this is a high correlative region and a low correlative region. This means that there is some sort of RNA that is present in both genes. Now for the result analysis, we can save the output as an image of the matrix. This image will get the suffix of TIF. We can also create a CSV document by choosing to output the matrix as a matrix of values. This can be viewed later in Excel. If you then want to examine a specific window, you can choose to write the sequence of a specific region to a .fna file. The header of the file will be that of the genome selected plus the desired genome or the desired sequence you wanted. You can also choose to BLAST the sequence. This will export the sequence and BLAST it with using BLAST-n with default parameters. The result will be written in a text file or a CSV file that you can open in Excel. Now you can also choose to analyze the unusual compositions. You can do that by choosing your average correlation and the max correlation a window can have, and then it will take all those windows and you can export it in a FASTA document. This will not be a single FASTA file, but a multi-FASTA file with all the regions, so windows that have the values that you want. The header will be given a genome label, the position window of the genome, and the values of the max correlation and the average correlation of that window. You can also use BLAST-n again to get the results back in a text file or a CSV file, but this is not recommended because you can have many windows that can have your parameters. There is also another window that will be not talking about here. That is an invalid window. That is when your window that you have selected, if 20% of that window contains letters that are not in a nucleotide sequence, so not A, G, A, T, G, and C, then this will be given a correlation value of 5, so it will appear pure black on your heat matrix. Then lastly, I will talk about the scientific question. So now we will analyze two different genomes of bacteria, and we will try to find out where there is a high region of correlation, and then we will see if the proteins that correspond to the translation of that specific sequence have something in common with each other. So let's do that. So first, we will go to our terminal. Now I already am in the folder where my S-plot is located, so you can do that too. Then we do Java, S-plot2jar, so now the program will open. We will be analyzing genome, so we select the S-plot by genome. Then we will first browse our first whole genome sequence. This will be Acidobacterium alveoli. So we can copy this and pose it as our X label. Now our other bacteria will be Vibrio cholerae. So we can also take this name and put it here. Now we will take a rather not too small but not too high k-mer size, because these two bacteria are not that alike. So we'll take a k-mer size of 5 and then a window size of 1250. No, we will take a window size of 2500 with 1250 offset. We will use our correlation labels and we will submit this. Now it will start calculating. We will also get a screen over here that will indicate us of how far the calculations have gone. So it has started. Now we just wait until it's finished. Now, we can see that our k-mer size was too big because we don't see much of correlation in here. So we will try that again. And we select S-plot by genome. Again, our two different bacteria. And we will take a smaller k-mer size, maybe 4, and then window size of 2500, offset of 1250, and submit this again. Hopefully this will give us a better result. Now this result is a bit better because now we can see a bit of correlation. Now I see a region that might be of some use for us. So we can zoom in. And here there is a region where there is a decent correlation. So over here we see that there is a bit of correlation diagonally. So we will now export the specific windows. So this is window 1976, 1978 of the Vibrio cholerae. So we will write that to a file. And we can name this file. I already have these files. I called it Vibrio here, over here, within the correlation, the average correlation. And then we can save that, but I already did it as a FASTA file. Now, I also did this for the Acido, and this was for window 2189 to 1191. So these windows. Now we will use these FASTA files and use NCBI BLAST. We will use BLASTX to translate the nucleotide sequences that we have to proteins. Now, because I was prepared, I already did this, and these were the outcomes. So we have the Vibrio over here. And then we have our acidobacteria over here. So for the Vibrio, we have a hexadeoxynuclease protein, and for the acidobacterium, we have a CRISPR-associated helicase endonuclease. Now, these two are very different proteins, but to answer our question, I think the correlation must have come because they are both nucleases. So that is the ending of my presentation.